This year will be the 30th anniversary of the Media Lab, and I was sitting around talking to actually one of our sponsors with Nicholas Negroponte, who was the first director of the Media Lab, and in a typical Nicholas sort of way, he said, you know, bio is the new digital. And he meant this at sort of multiple levels, which I think is really interesting, because if you think 30 years ago when we created the Media Lab, there was no Macintosh. It was sort of the age of Apple Computer rising. It was actually, Nicholas had written a book called Being Digital. I remember when people used to call themselves, I'm a digital kind of guy, you know. And so if you think about all the different companies and how much the world has changed since then, and so, and I started probably being more actively involved a little bit later. This is a picture of my bathroom in the early 90s. It's the first this is the first commercial internet service provider in Japan. And it was when you could take a bunch of junk parts and throw together an ISP. And this has a couple key points. It's permissionless innovation. It was a forced competition that allowed us to drive the cost of communication to nearly zero, which allowed collaboration and distribution to also become nearly zero. And you throw into that mix Moore's Law, which you all know, and then what you get is you get low cost of networks and low cost of computing, creates an abundance of free and open source software that fundamentally changes the cost of innovation. And so what this does is, if you remember, Boston used to be big with computers, and there was this regional shift from Boston to out here in Silicon Valley, and a lot of it had to do with the fact that when it costs a lot of money to do everything, you have a bunch of people wearing neckties sitting around making plans so that they can then invest the money and then hire the engineers. I call this Mba Driven Innovation. But what happened once you got the internet, once you had Linux, once you had all of the stack basically nearly free, is that you could make a Google, Yahoo, or Facebook without any money. All of these products were created initially without anything other than a couple of engineers and designers getting together, deciding to do it, and then later you raise the money, and then maybe you need somebody with an MBA before you go public. Nothing against people with MBAs. But you don't have them at the beginning. And so what happens is it pushes innovation from centralized power to the edges, right? So we all know this story. This is the story of the internet. This is the story of venture capital. But it turns out it's happening in hardware, and this is what a lot of Solid is about. So this is one of our Geiger counters from our Safecast project. But if you look inside of this Geiger counter, there's tons and tons of open stuff. There's Arduinos. We didn't actually have to reinvent much to make this. This case is a standard Pelican case, and this is the open hardware mark. So it turns out it's not exactly the same, but sort of similar to what happened with software and open source. You see that happening now in the hardware domain, so you don't really have to reinvent every single piece of hardware from scratch. You actually don't even have to reinvent the factory. This is Limor from Adafruit, one of our graduates. They're going to be doing a session. But she's standing in front of a Samsung pick-and-place machine, which is basically a factory in a box. You can buy this on the internet. This is her favorite gadget, she says. Or you can go to Shenzhen, which is basically the Silicon Valley of hardware, where everything is happening. Unfortunately, a bunch of my students are over there with Bunny, and so they can't be here today, but they are immersed there. And so when you go to Shenzhen, what happens is everything is so fast and so iterative that kids make cell phones in sort of the same way in Silicon Valley people make websites and apps. It's sort of this agile, emergent craziness. And so what's happening in hardware is what happened in software, which is innovation is getting pushed to the edges because the cost of innovation is getting so fast and so cheap. So this is biology. This is Sorona, which is a biological process using a synthetic microbe that converts sugar into polyester at about 30% higher efficiency than the traditional fossil fuel method. The problem is it took years and hundreds of millions of dollars to develop this microbe because it was very expensive, very difficult to do. What's happening is Moore's Law, which was the reason Moore's Law works is you use computers to design the chips, use mass manufacturing to create their wafers, and you're able to reduce the cost of computing so fast, doubling every 18 months. This is now happening in synthetic biology, right? So you can now take all of the gene bricks, these little parts of genetic code, categorize them as if they were pieces of code, write software using a computer, stick them in a bacteria, reboot the bacteria, and the bacteria, just as with computers, usually does what you think it does. So this is an example of some work done in our lab by a student named Charles Fracchia. But this is an example. So here's a synthetic piece of life. It's got a genetically engineered plasmid inside. This little hook thing is a promoter. It's looking for some, I think, glucose in the environment. When the glucose is sensed, the promoter triggers and expresses a gene that then takes potassium and sends it out into the environment. Then he's got an electrical sensor that senses the change in voltage and then sends a wireless signal to the computer so you know when there's glucose in the environment. You could just swap out that biobrick of the promoter to some other material sensor, like sensing arsenic, and so you could change this into an arsenic sensor. The key here is that for him, biology and electronics is fungible. You could have done this in solid state electronics, you could have done this in biology, but this is lower power, more stochastic. This needs to be fast and effective. So this is a thing that he used. But what's happening here is biology is being mashed together with electronics in a seamless way. This is Joe Jacobson, who will be speaking later today. He's one of our faculty members. He is now printing genes on a silicon wafer. So you used to have to put these things together in pipettes by hand. You can now print them on a gene printer. So Illumina is one of the big companies in this field. If you remember, in 2003, it cost $3.5 billion or $2.5 billion to sequence the human genome. Now, on this machine, it costs about $1,000. And then last month, Nanopore, Oxford Nanopore, just came out with this amazing device. This is a real thing. This is a $1,000 gene sequencer that was deployed in Africa recently to sequence, look for mutations in Ebola. So now what you've gone is from billions of dollars in 2003 to a $1,000 handheld USB gene encoder. When you talk to George Church, he'll say that biology is being reduced in cost by about 5 or 6 times the speed of Moore's law. It's coming faster than the internet. This is another company that I've recently gotten involved in. So Emerald Therapeutics has this very interesting thing in the cloud. These are a bunch of kids who were writing code to help take experimental data and standardize it and visualize it using Mathematica and other tools. But they said, well, why can't we control the lab itself? And so they created a language to control biological experimentation devices and start using robots. So what you can do now, because as you heard, science is sort of broken because people can't reproduce each other's experiments. What if all the experiments were conducted by robots and you had a cloud of a wet lab, so you didn't actually need a wet lab, and if somebody wanted to question an experiment, they could go audit it, they could take your data, and they could change one piece by 3 degrees and run the experiment again, and you could reproduce it. So this is starting to standardize the experimentation system, also reduce the cost. But the thing that's interesting, when you look at a lot of these companies, the big synth bio labs, I see Connor from Symbiota there, but a lot of the people who are running things in synthetic biology and biology are not the biologists. They're mostly ex-computer science and robots people. So this is another part of the convergence, I think, is that the people are crossing over. This is with some of the help with Symbiota and some others, this is us designing, putting the genes together, inserting it into a bacteria and rebooting a bacteria to create our own form of life, a new bacteria that creates violacin in our kitchen. You know, a couple decades ago, this would have been a Nobel Prize. Now, you've got hundreds of people doing this in their kitchen. This is called street bio, or kitchen bio. iGEM is a spin out of MIT. It's the International Genetically Engineered Machines competition where thousands of kids, including high school kids, get together, and instead of building robots, they build synthetic life together, like bacteria that smell like bananas or bacteria that may search for landmines. George Church was recently on the Cobell Report where he had encoded his book into the DNA of a bacteria and allowed it to reproduce 20 million times, so he could give 20 million copies to Cobell. But what's important is when you look at the archival density of the genes themselves, he thinks that you can put 200 million terabytes per gram. That's much more dense than a hard disk, and that it might last for 700,000 years. So actually, this hasn't been officially published, but it was in the annual report. So I'll say it, Technicolor has contracted him to start thinking about doing video archival in DNA. So it's not, you can imagine sensors and memory also becoming. So George will say, why are we sitting here trying to make computers in silicon be like human brains in artificial intelligence? Why don't we just make brains smarter and be more like computers? This is some work by Neri Oxman in our lab. So there's a bacteria called cyanobacteria that converts light into sugar, and she combined it together with E. coli that converts the sugar into biofuels. And she wanted to come up with a place where we can store this stuff to allow this thing to work properly. So she designed an algorithm based on something that looks like our intestinal system, and then created this sort of wearable 3D printed, this is using Stratasys technology, and it has the bacteria inside that flows, so it's a bacterial environment. This is sort of a prototype for an idea of a wearable system that has a biological, synthetic biology ecosystem inside that might create some sort of fuel system. But the idea here, and this is where I'm going to end, is this last slide, is so this is the way Neri's brain is structured. So she works on computational design, which is using algorithms to design things. So when you have, instead of building a building and then trying to compute the biological environment, you take the biological environment and use computation to compute the building, compute the structures, and you add to that the ability to actually print these things through additive manufacturing, the fact that you can now modify the environment itself, the bacteria and other things in the environment through synthetic biology, and that we have new materials, and we'll hear a lot about these new materials throughout the conference. And you bring this together, and this is a new kind of design. You had industrial design, you had Tim Brown's design thinking. This is synthetic biology, computational. This is the design of the future. And the design of the future is hardware, it's software, it's biology, and it's complex. But you have to know math. So hopefully we will see a lot of this at the rest of the conference. Thank you very much.